Think about this for a second. How exactly do you kill a pathogen that survives by literally hijacking your very own cellular factories? In our previous explainer, we looked at how animal and bacterial viruses replicate, how they break in and take over. But that leaves us with this massive biological problem. How do we eliminate a rogue virus without destroying the human host right along with it? If you're heading into nursing, med school, or any allied health career, figuring this out is basically the holy grail of infectious disease control. By the end of this explainer, you're going to understand exactly how we grow these invisible enemies in the lab, how we detect them when they breach a patient's system, and the absolute genius pharmacology we use to take them out. Here's our roadmap for today. We're going to cover cultivating the invisible enemy, look at the global viral toll, dive into clinical forensics, tackle the antiviral hurdle, and wrap up with smart laser solutions. All right, let's jump straight into part 1, cultivating the invisible enemy, basically our laboratory greenhouse. So because viruses are obligate intracellular parasites, you can't just swipe a sample onto a standard agar plate and expect little colonies to pop up. They need living host cells, right? We cultivate these pathogens in the lab for 3 big reasons. First, isolation and diagnostic identification, basically figuring out exactly what's making the patient sick. Second, vaccine development, so we can mass produce preventative defenses. And third, basic scientific research, just to understand how these viral machines actually tick. Now, if we compare our 3 main cultivation platforms, they each have really specific medical uses. First up is cell culture. This is an in vitro method where we grow a controlled layer of living cells in a dish, mostly for diagnostic isolation and looking for tissue damage, which we call plaques. Then we have bird embryos. These act like tiny, self-contained in vivo incubators. We actually micro-inject viruses into them to mass-produce stuff like our annual flu vaccines. And finally, live animal inoculation. This uses specialized models like lab mice to study systemic pathogenesis, basically seeing how a whole living immune system handles the infection. Moving on to Section 2, the global viral toll, a biosphere-wide data invasion. Let's zoom out for a second and look at the sheer scale of this biological impact. We're dealing with high-volume acute infections, think rhinoviruses that cause literally billions of mild colds every single year. But then we also face severe pathogenicity. These are highly virulent viruses like Ebola or rabies that cause catastrophic system failures and, unfortunately, really high mortality rates. We're also seeing that some latent viruses are strongly linked to chronic afflictions down the road, things like autoimmune disorders or even oncogenic or cancer-causing transformations. But hey, it's not all bad news. Ecologically speaking, viruses actually provide essential ecosystem stabilization. They're out there regulating microbial populations and helping recycle nutrients across the entire biosphere. Okay, Section 3, clinical forensics, detecting the invisible enemy. Let's put our clinical hats back on. When a patient comes in, how do we diagnose a systemic breach that we can't even see? Well, step 1 is the overall clinical picture. We look for distinct physical signs like a shingles rash or a specific pattern of fever. Step 2 is direct antigen and PCR screening. This is where we scan for physical viral fragments or amplify the virus's genetic code so we can actually see it. Step 3 is serological antibody screening. Here, we're checking the patient's blood serum to see if their immune system has manufactured specific antibodies tailored to fight that exact threat. And finally, step 4 is cell culture isolation, where we literally watch the cytopathic damage unfold on cells under a microscope. Which brings us to Section 4, the antiviral hurdle, the collateral damage nightmare. So once we've actually identified the virus, we hit a massive pharmacological wall, a low therapeutic index. Remember, viruses completely hijack our own eukaryotic ribosomes and metabolic pathways to build their copies. And because of that, if you try to use a broad chemical weapon to shut down the virus, you're probably going to severely damage the patient's healthy tissue right along with it. The line between killing the invader and poisoning the host is razor thin. But enter Section 5, smart laser solutions, achieving selective toxicity. Thankfully, modern pharmacology has come up with a brilliant workaround. It's called selective toxicity. This is absolutely the holy grail of antiviral medicine. It's the ability of a drug to knock out the virus while causing basically zero harm to healthy human cells. And how do we do that? By designing drugs that specifically target unique viral enzymes or life cycle steps that human cells just don't have. Let's look at 2 incredible real-world examples of this. On the left, we've got Acyclovir, brand name Zovirex. I like to think of this one as the viral sleeper trap, used mostly for herpes and varicella zoster outbreaks.
And over on the right is oseltamivir, which you probably know as Tamiflu. This one acts as a release blockade for seasonal influenza.
Let's break down Acyclovir first, because this mechanism is just stunning. It's administered as what we call a prodrug, meaning it's totally inactive at first. It just wanders freely through the body, and healthy human cells completely ignore it. But the second it enters a cell that's infected by the herpes virus, the virus uses its very own unique enzyme, viral thymidine kinase, to activate the drug. The virus literally arms the bomb itself. How crazy is that? And once that bomb is armed, you get immediate chain termination. The newly activated drug jams itself into the viral DNA strand and just instantly breaks the printing press. The whole viral assembly line completely collapses, and because human cells don't naturally have that specific activating enzyme, uninfected cells are perfectly safe. It's just a masterclass in selective toxicity.
Now let's look at oseltamivir, or Tamiflu. When newly formed influenza viruses are ready to bud off and escape a host cell, they use a specialized surface enzyme called neuraminidase to basically cut themselves loose. Tamiflu acts as a highly specific structural clamp that binds right to that viral escape hook. With the hook totally jammed, the newborn viruses are permanently tethered to the outside of the dying host cell. They're stuck, meaning they can't spread further down into the respiratory tract.
And the reason this works so well without hurting the patient is that human cell logistics just do not use the neuraminidase pathway, period. Because we're targeting an exit gate that our own cells don't even possess, Tamiflu remains incredibly safe for host tissues. It's a highly targeted defense patch for our cellular metropolis. So, achieving selective toxicity by targeting unique viral machinery is an amazing triumph. But what if we have to fight a pathogen that doesn't even have genetic code to target in the first place? That is the terrifying reality of prions. Go ahead and jump right into the next explainer in this playlist to uncover how these rule-breaking infectious proteins destroy the brain. I'll see you over there.